Imagine taking a living bacterial cell, like E. coli, and physically grinding it down into its raw molecular parts. You have all the carbon, hydrogen, and complex proteins required for life, just mechanically separated. If you place those exact molecular components back into a nutrient-rich bath, they do absolutely nothing. The chemistry sits inert. Compare this to an experiment with the artemia, or brine shrimp. If you freeze these organisms to absolute zero, halting all chemical processes, they resume their normal life cycle the moment they are thawed. Both experiments stop life, but physical grinding destroys the internal chemical organization, while freezing merely pauses it. The molecules are identical in both cases. The arrangement is what changes. This tells us that life is an emergent property of chemical organization and interaction. It is not dictated simply by the presence of specific biological molecules, but by how those molecules interact. When looking at biological entities, we see a complex system. In ant colonies or brains, thousands of simple local interactions create sophisticated system-wide behaviors. This exact same dynamic applies at the molecular level. On the early Earth, the primordial soup was a chaotic, undirected environment full of simple organic components. A persistent assumption in origin of life research is that we must find a single, highly complex molecule, like RNA, that managed to catalyze its own replication by sheer chance. The math and the chemistry point to a different mechanism. Life emerged through autocatalytic sets, networks of incredibly simple molecules that acted collectively. Interconnected chemical networks functioned as the primary units of evolutionary selection, long before individual molecules could replicate. To understand why a network is necessary, we have to look at the primary thermodynamic problem facing the early Earth. Natural organic reactions are too slow to sustain anything resembling life. This chart illustrates the scale of the problem. It compares the rates of organic chemical reactions with and without catalysts. Look at the vertical gap. Reactions at the bottom, taking millions of years naturally, happen in fractions of a second when catalyzed at the top. A catalyst is simply a molecule that accelerates a reaction without being consumed by it. Without them, the chemical synchronization required for biology is impossible. This creates a self-referential paradox. Life requires catalysts to exist, yet in modern biology today, complex catalysts like enzymes are only manufactured by systems that are already living. This paradox makes the spontaneous generation of a single, complex, self-replicating molecule highly improbable. A dead ocean cannot produce the very enzymes required to build the first biological factory. To overcome this kinetic barrier, prebiotic chemistry had to find a way to produce its own catalysts, using only the simple, non-biological materials at hand. This diagram shows one possible mechanism: simple autocatalysis. Molecule C directly accelerates its own creation by catalyzing the reaction of A and B. While simple autocatalysis exists, it is chemically rare, lacking the robustness to jumpstart an entire biosphere. The alternative is shown here: coupled autocatalysis. Instead of a molecule making itself, the labor is distributed. Molecule C catalyzes the formation of E. In return, molecule E, or another product in the chain, catalyzes the formation of C. When you expand this to a massive scale, you get an entire network where no single molecule makes itself. Instead, the molecules mutually catalyze each other's formation from a basic food source. Once a closed loop forms, the entire system exhibits exponential growth. This mathematical signature directly mirrors the explosive growth seen in living populations. Self-replication is an emergent property of the network's topology. By working together, incredibly simple molecules collectively achieve what a single replicator cannot. Proving that these networks exist requires finding specific, self-sustaining loops hidden inside massive, randomly simulated chemical environments. Researchers use a mathematical tool called the RAF framework, which stands for Reflexively Autocatalytic and Food-Generated Networks, to detect these systems. This is a directed graph model of a chemical reaction network. It translates raw chemistry into nodes and edges. The outer unbolded arrows represent the food set, raw materials feeding the system. The bolded central loop, connecting E1 through E15, is the viable core. This is the exact place where the catalytic sequence closes on itself and becomes self-sustaining. By analyzing the adjacency matrices of these networks using linear algebra, algorithms can consistently detect these hidden cores within massive, randomly simulated chemical environments. The emergence of these cores shows that the origin of life is not a chemical accident. It is a mathematically predictable phase transition that inevitably occurs when a network of reactions reaches a critical density. This mathematical model solves one of the oldest challenges in prebiotic chemistry, the mixtures problem. A random primordial soup usually produces chaotic, unusable tar rather than organized biology. Autocatalytic networks operate like chemical vacuums. They selectively consume raw materials to amplify specific pathways, starving out random side reactions. To maintain efficiency, the network requires physical containment. Spontaneously forming structures, like lipid vesicles, act as primitive compartments to isolate the network from external noise. If multiple distinct autocatalytic cores end up trapped inside the same lipid compartment, they must share the same incoming food supply. Environmental factors, like a slight temperature fluctuation, will dictate which core operates faster. The more efficient network consumes the shared resources, suppressing the other. This internal competition for resources within a closed compartment established the first functional natural selection, governed strictly by chemical kinetics rather than biological genes. If these networks are subject to Darwinian selection, how do they pass on traits or mutate without a genetic code like DNA or RNA? The answer lies in the compositional genome. The identity of an autocatalytic network resides in the specific ratios and connectivity strengths of its internal pathways. The random insertion of a new molecule or a shift in the environment alters these pathway strengths. This causes the entire network topology to shift. This topological shift is the prebiotic equivalent of a genetic mutation. It completely alters the network's behavior and its efficiency in processing food. Through this mechanism, these chemical systems were able to adapt, grow, and divide. They were evolving long before the first sequence-based biopolymers emerged. The essence of life has always been the network. It is a beautiful, self-sustaining mathematical architecture that transformed dead matter into the engines of biology.